right, guys, so first of all, thank you for joining us for this presentation. What I'm going to be discussing is some of the research that my lab has been working on over the past 4 or 5 years, which really looks at trying to understand the extent of biological invasions in a region known as the Adirondack Park. All right, so for those of you who don't know, the Adirondack Park is a region located in northern New York, so about 7 hours drive north of New York City. So you can see this area right here. And it's actually the largest protected forest reserve in the contiguous United States. We have quite a lot of lakes and rivers, and there's been a lot of work in terms of understanding the water chemistry of the Adirondacks. And that's because compared to many of the other lakes in North America, the lakes of the Adirondacks are notoriously acidic, which makes it a really interesting place to study molluscan diversity. Now, when I first came to the Adirondack Park, just to give you a background in terms of how I got into this area of research, I was essentially came in as a marine biologist, right? So I worked a lot on polychaetes, particularly polychaetes that parasitize oysters. So I was an annelid and a molluscan person. And when I came to the Adirondacks, one of the things that I wanted to do is I wanted to find a species of mollusk, whether it's a bivalve or a gastropod, to use as a model species in my lab to do some really cool developmental studies and genetic studies. And one of the things I did was I found the freshwater snails of New York State, which was written by Dr. Eileen Jokinen back in the early 90s. But essentially, this is actually one of the most comprehensive surveys or regional surveys of gastropods that you can find anywhere else. All right, so over the last couple of years, these are the different areas that my students and I have sampled. So we covered quite a lot of ground, both at the foothills of the Adirondack Park, near the St. Lawrence River, and also deep within the Adirondack Park. But even though, you know, it seems as though it's a lot, it's sort of a drop in a bucket of water, we still have a lot of unexplored areas of the Adirondacks we want to look at in terms of looking at molluscan diversity. Now, the first thing we noticed, or I noticed, was that many of the snails we were collecting, we can find in Dr. Jokinen's surveys. In fact, the zebra mussels, well, first of all, that's a bivalve, so we're not going to find it there anyway. But the thing we noticed was that even with the bivalves and the gastropods, everything was basically invasive, right? So we have the so-called mystery snails, right? So the Chinese mystery snail, the banded mystery snail, the New Zealand mud snails. And so what we found was that some of the species that Dr. Jokinen recorded in surveys, we can actually find when we sampled specifically the same rivers and lakes that she sampled, right? And so what we think happened is that there was a change in the composition of mollusks over the last, 20 plus years, 20 years or so. And so what I'll be talking about briefly is some of the new records of non-native gastropods that we found in the Adirondack Park. Well, one particular new record. Then I'll be looking at some of the experimental stuff we've been looking at in terms of seeing how these invasive gastropods respond to environmental stresses. And then I'll be mentioning some of the work that is ongoing right now in my lab, which is looking at connectivity of invasive mollusks in the Adirondack region. And this work is really being spearheaded by my doctoral student. And it actually officially begins this summer, even though we've been developing genetic markers right now. So one of the first studies that we did in this Adirondack region was a typical rapid assessment survey where we would go out into the lakes and rivers, sample for an hour at a specific depth, and collect as many mollusks as we can, gastropods and bivalves. And Sarah Coates, who was a McNair student, and Hannah Baby, these were the two students that really spearheaded a lot of the field work. We would bring the snails back into the lab. We'd take a lot of these different conchological characters. We would borrow specimens from the New York State Museum, which there's a lot of because of Dr. Jokinen's work, and the Smithsonian, and we would try to key out these mollusks. And one of the things we noticed was that there's quite a lot of polymorphism that existed within all of these invasive mollusks that we were able to collect. The first one was Cipangopaludina chinensis, also known as the Chinese mystery snail. Now, this particular snail, we've known in terms of its distribution in the Adirondacks for quite a long while. The Japanese mystery snail was thought to have been present in the area, but a lot of researchers found that at the time that people were misidentifying it, and there is just one species. And we were we were thinking there's probably more than one species we're dealing with. And so what we did was we keyed out species that we thought were Cipangopaludina japonica. We compared it with specimens from the type locality using conchological characteristics, and we found very, very close similarities. Then what we did was we did our genetic analysis, so we barcoded the specimens we thought was C. japonica and the C. chinensis specimens. And what we found was that there were two distinct clades where each of these species eventually nested into, right? So we were able to confirm reciprocal monophyly of both of these specimens with sequences from the type locality, right? So we did confirm that C. japonica is in fact present in the Adirondack Park, and it occurs, its range overlaps with C. chinensis. In fact, you can find both snails, together in the same rivers and in the same lakes. Now, what's really interesting about this discovery was that C. japonica is also present in Texas. It's been genetically confirmed in the lakes in Texas. And this gives you an idea of how sort of broad the thermotolerances of these mystery snails, right, quote unquote, right? The mere fact that C. japonica can survive in the lakes of Texas, where it's obviously warmer, compared to, appearing in the Adirondacks, where the lakes basically freeze over for 4 or 5 months out of the year. So the fact that they can survive in both of those environments gives you an idea of how broad their thermotolerance is. Okay. So with that being said, one of the projects that we've done, or I've been interested in, is trying to figure out, well, how broad really is their tolerance to environmental stresses? So Megan and Lily, two environmental science majors, one of them has graduated already, they began looking at the effects of carbon dioxide-induced acidification on one of these invasive mystery snails, particularly Viviparus georgianus, right, the banded mystery snail, because you have these chestnut-colored bands that you can find on the shell, right? Now, there's been a lot of work done on CO2-induced acidification on marine gastropods, but till now, or till this study came out, there was none that was done on freshwater gastropods. And so, what we did was we created an experimental setup for freshwater habitats. And you can sort of see what the setup looked like, right? So this is our CO2 tank, which pumps carbon dioxide into our CO2 reactor, which eventually we can regulate the pH, and then out pumps the water, the pH that we specified, right? And so what we did was we measured the survivorship of snails that were cultured under control, particularly 7.3. We dropped the pH to 6.8 and then to 6.3 again. And one of the things we found was that after 12 weeks, there was no change in survival. So all of these snails survived those wide differences in pH. There was no change in the growth rates, as you can see from the scatter plot. And also, what we did was we decided to make incisions in the shell so we intentionally damaged the shells of these snails. And we looked at shell repair as a proxy for physiological performance. And surprisingly, at the 7.3 and 6.8 treatment, the snails did pretty well. They all repaired their shells after 12 weeks. It was only at the 6.3 treatment that about 20%, only 20% of the snails were able to repair their shell. And so it gives you an idea of how, tolerant these snails are of low acidic environments. And it gives you an idea, it makes sense why they would be thriving in these highly acidic lakes of the Adirondacks. And so the research that we're doing right now is trying to look at genetic connectivity in the Adirondack Park using invasive snails as our study species. So my PhD student is working on the Raquette River, which you can see over here. It's the second longest river in New York State. It flows into the massive St. Lawrence, which eventually empties into the Atlantic Ocean. Right? Now, what makes the Raquette particularly interesting is that it's actually one of the most dammed rivers east of the Mississippi. It's one of the most anthropogenically disturbed rivers in the Northeast, right? And as you know, dams can act as dispersal barriers. But what makes this story even more interesting is the fact that in the Adirondacks, we have problems of a lot of recreational fishermen and boaters and these mechanisms can act as vectors that can promote dispersal. So on one hand, we have potential anthropogenically mediated gene flow, and then we have anthropogenic dispersal barriers. So trying to disentangle the connectivity patterns of invasive snails in this type of environment is something that we're really interested in and something that we're really excited about doing over the next couple of years. So to acknowledge, as all of you might have seen, a lot of these projects have been driven by undergraduates' participation. And so, they've actually played a very big role in terms of helping push a lot of my projects forward. Funding the St. Lawrence River Research and Education Fund, which is part of the New York Power Authority, played a big role in funding a lot of these initiatives, and also the Malacological Society of London. So thank you. And if you have any questions, just let me know.